Hello, I'm Juan Jose Monterrey Martinez, and I am doing Chapter 11, Meiosis. Okay, key structures that we're going to be talking about are the chromosome. So here on the bottom left, let's say this blue chromosome is coming from my father, and the red chromosome is coming from my mother. Now, together, if I were to link these together, this would be considered, let's say, chromosome number 1. Because remember, chromosomes are two sets of genes. So, I have all of my genes from my father on the blue, but I still need to have a matching, genetically identical gene allele, which would be from my mother, which would be the red. So they are kind of complementary, kind of like a shoe, right? So you have your left and you have your right foot, and together you have a pair. So I have one genetic shoe from my father and one genetic shoe from my mother, and these are homologous pairs. They connect, but they also connect in a very unique way that sets it apart from mitosis. Another feature to point out is that mitosis mitosis divides only once, as opposed to meiosis will divide twice. To give a better explanation of what that division means is that here at the bottom we have mitosis, in which at interphase we are duplicating our chromosomes, and then through prophase, metaphase, telophase, we have created a new identical cell, which is what we would want if I cut my finger and I needed to replicate my cells to be identical. However, when it comes to our sex cells, we don't want identical cells. We want as much genetic variation to occur. And the way that we are able to achieve that is that when meiosis 1 occurs, we've divided once, but as we look further into those homologous pairs that I referred to earlier, there's been a recombination, and that recombination then requires another set of cell division, and that is meiosis 2. So in meiosis 1, we have a linkage of those homologous pairs, and then in meiosis 2, we have a continuation of division, so that by the end, we have as much genetic recombinations in our sex cells as we can possibly. So we want a lot of genetic variation in our sex cells. So going back to those homologous pairs, I want you to envision it like a Velcro. You have one set of genetic material, and then you have the other set, and they come together at a synapsis, and this is a kind of like a Velcro. They hook onto each other, and that's what you're seeing here at the on the diagram, where it's just kind of like those white finger-like extensions. Those are snaptonemal. That's a septonemal complex. They are kind of these protein structures that bind one chromosome from my father's side to my mother's side, and then they are kind of linked in together. And the point of that protein complex there is that it gives some structural support for genetic variation to start, that I was saying here. Now, there isn't just like this free-for-all in which genetic material can be exchanged at any point in the chromosome. There are kind of these large protein signalers called recombination indicators that say, hey, I want genetic material exchanged here for the purpose of promoting that kind of genetic diversity. Okay, so to begin meiosis one, we're going to start by replicating a bunch of our genetic material, and then in all of that genetic material is going to recombine, and then that's where the homologous pairs will find each other. Those homologous pairs will link to each other. Once they've linked, then they will do their crossover, and then as the meiosis one starts to separate through prophase, metaphase, anaphase, you really start to see those areas that did have some recombinations start to pull apart, and that's the point because we want to have two daughter cells that have some genetic recombination. So following from meiosis one, we have two new daughter cells that have those uh, chromosomes that have those recombination areas, but we can't stop there. That's too much genetic material, and the whole point of meiosis is to create as much genetic variation as we can. So with those two new haploid cells, we're going to want to continue the division. So we'll go through prophase 2, metaphase 2, anaphase 2, telophase 2 to to be able to have 4 different daughter cells of genetic material that is all recombinated. And this picture might do a little bit better where you see on the bottom right, those are 4 different genetically different variant cells, and that's what we optimally want. We want as much genetic variation in our sex cells to evolutionary be diverse. Thank you very much.